Hello all. We have covered sterility assay and your microbial limit test till now. Today we are going to cover the last part of this particular chapter, that is your microbiological assay. Now what is microbiological assay of an antibiotic? Now we all know what is the function of an antibiotic. The function of an antibiotic is to kill the microorganisms. So that means that when you perform the assay on any antibiotic, the main criteria that you will be selecting to evaluate an antibiotic is how efficiently that particular antibiotic is able to kill that particular strain of microorganism. So basically in all your microbiological assay, what you are going to do is, for example, you have developed a new antibiotic in the market, or you have introduced a new antibiotic in the market. So to check whether your antibiotic is at par with the already available antibiotic, you are going to perform a microbial assay. So basically you are going to compare the antimicrobial activity of your antibiotic against the antimicrobial activity of the already available antibiotic. So it says that inhibition of growth under standardized conditions may be used as a method for demonstrating the therapeutic efficacy of your antibiotics. Even a small change in the structure of the antibiotic molecule can lead to a vast difference in its antimicrobial activity, and hence microbiological assays are very useful for resolving doubts regarding possible changes in the potency of the antibiotic and their preparations. So what is the principle behind your microbiological assay? It's actually based upon a comparison of the inhibition of growth of microorganisms by measured concentration of the antibiotics to be examined with that produced by known concentrations of a standard preparation of an antibiotic having known activity. So what this complicated principle means, in this what you are going to do is, you are going to select your antibiotic and you are going to compare this selected antibiotic with an already available antibiotic with having a known activity. Okay. Now during this selection, it is very important that the concentration that you use for your antibiotic and the concentration that you will be using of the known antibiotic should be same, not almost, should be exactly same. That means you should be comparing both the antibiotics at the same concentration. Okay. Now there are two methods for doing your microbiological assay. First method is called as your cylinder plate method, which is also called as your cup and your plate method, and second method is your turbidometric method, which is also called as your tube assay. Now we'll see what these methods mean. This slide basically gives you an idea how to make your media. So basically you take all the ingredients in sufficient water and you make 1000 ml, and later on the pH is adjusted by using sodium hydroxide or hydrochloric acid, so that after sterilization the pH is between 6.5 to 7.5. This slide gives you an overview of how to prepare buffers, prepare buffer solutions. Now basically buffer solutions are used as diluting solutions. Now the antibiotic that we are going to test, we are not going to use it in the same concentration. We are obviously going to dilute it so that not all the microorganisms in a particular petri dish are killed because of high concentration of antibiotic. So that is why appropriate dilution is required. This dilution is done with the help of buffer solutions. So you can see here there are different types of buffer, right from buffer number 1 to buffer number 6, which has different concentrations of different salts such as dipotassium hydrogen phosphate and potassium dihydrogen phosphate. And later on, after sterilization, you need to adjust the pH with either phosphoric acid or potassium hydroxide to get the desired pH that is mentioned in the table. Now this slide basically tells you how to make your stock solutions and dilutions of your test solution. So basically these are the antibiotics. In the first column you can see are the antibiotics of which we know the activity. So they are your standard antibiotics such as your amic acid, amphotericin B, bacitracin, bleomycin, etc. The second column tells you which method you should use if you are working with that particular antibiotic. So method A is your cup and plate method and method B is your turbidometric method. Column 3 tells you whether you require drying or not. Then column 4 tells you which type of solvent you should use for initial dilution and what should be your stock concentration. Okay. Similarly, the next table tells you what should be your dilution or the dose that should be present for the particular test substance. Okay. Now this slide gives you an idea about preparation of the test organisms. Now for each antibiotic, the test organism that needs to be used is also fixed. So for example, if you are using amic acid as your standard antibiotic, you are going to use Staphylococcus aureus. So ATCC is a kind of a library in which all kind of microorganisms are available and they are given a specific number. So if you are referring to ATCC library 29737 will always denote that the strain of the microorganism or that particular microorganism is your Staphylococcus aureus. Okay, now let's see the first method, and that is your cylinder plate or cup plate method. Now you all know how a petri dish that is filled with nutrient agar looks like. So basically it looks like a semi-solid jelly-like structure which is kept inside a petri dish. Okay. Now we are going to take this petri dish, and inside this petri dish we are going to bore some holes in it with the help of a borer. So basically you have, now this process you have to imagine it because you cannot see it obviously. So you have a petri dish in which you have filled your nutrient agar up to a particular height. Okay. Now when the gel solidifies with the help of a borer is basically kind of a hollow cylinder. Okay. So with the help of this hollow cylinder you are going to bore holes inside the petri dish. If the petri dish is a bigger petri dish, we usually bore 6 holes in this petri dish. If the petri dish is a smaller petri dish, maximum 3 holes are bored into the petri dish. Okay. So now let's move to the name. That is why this method is called as your cup and plate method. So whatever holes you have created in the petri dish becomes your cup, and your petri dish itself is called as a plate. Hence the name is your cup and plate method. Okay. Now what are we going to do in this cup and plate method is, I'll tell you the entire procedure. Okay. So initially we take the petri dish and we keep it in an autoclave for sterilization. Okay. Now we remove the petri dish and take it into the aseptic area. Now we take the nutrient agar which is still hot, that means it is still in the liquid state. This is present in a conical flask. Okay, I repeat, you have a nutrient agar which is hot, which is in the liquid state, present in a conical flask. Okay, in this you add the selected microorganisms. Okay, this can be say 0.1 ml or even 0.5 ml. not more than that. That dilution you need to calculate according to how much is the concentration of microorganisms in the stock solution. Okay. Now to this conical flask you add the microorganisms, you stir it, and you pour this liquid nutrient agar in your already sterilized petri dish. Okay, and you allow this petri dish to solidify, or rather to cool down, so that ultimately the nutrient agar gets solidified. So what you have basically done is you have used a pour plate technique. I again tell you what is a pour plate technique. In this, microorganisms are uniformly distributed throughout the media and then it is poured onto the plate. So the microorganisms are on the surface as well as they are submerged. Okay, so now our petri dish, which contains microorganisms on the surface as well as submerged, they are ready. Now this petri dish, we are going to take that borer, which is nothing but a hollow cylinder, and we are going to bore holes into the petri dish. Okay, so these holes now act as our cup, and your petri dish now itself becomes a plate. Okay, now suppose for the simplicity, you have made two holes in the petri dish. Okay, so what you are going to do is, in one of the hole you are going to pour your antibiotic. That means the antibiotic that is newly developed or which is not yet in the market. And in the second hole you are going to pour the same concentration of antibiotic, which is the activity you already know. Okay, so I repeat, there are two holes. In one hole you have put your antibiotic which is to be studied, and in the other hole you have put an antibiotic of the known activity. Both are used in the same concentration. Okay, now this petri dish you are going to keep it in your incubator for say 24 hours. Okay, now after 24 hours what you observe is, obviously the work of antibiotic is to kill the microorganisms. Okay, so after 24 hours you will see that around that cup you will see a region of you will see a clear region. Okay, now why the region is clear around that particular hole in which you have put antibiotic is because your antibiotic has migrated from the hole into the nutrient medium and it has killed the microorganisms that were there around the hole. Okay, so this particular region where there is no microorganism present inside your petri dish is called as zone of inhibition. ZOI, zone of inhibition. So that zone is basically the zone that is created because the antibiotic has killed the microorganisms in that particular area. Okay, now it is very obvious that if the antibiotic is more active, that means it has more activity, your zone of inhibition will be bigger. Okay, if your antibiotic has lesser activity, your zone of inhibition will be smaller. Okay, so if you are comparing two antibiotics, one is your test and one is your standard, if the zone of inhibition of both the antibiotics is same, both has same activity. If the zone of inhibition of your test is smaller than your standard is a better antibiotic, but your test is not a better antibiotic. Okay, so this is how we can use cup and plate method to find out assay of an antibiotic.